This conference will now be recorded.
Okay. A very good morning, everyone. Welcome to the classes of applied microbiology. So today we have applied microbiology for Klebsiella. Klebsiella chapter has already been taught to you in Enterobacteriaceae. You have already seen the plates of Klebsiella. Also, you might remember how much mucoid it was. It is a very mucoid colony which is produced by Klebsiella pneumoniae on blood agar, be it McConkey, be it mutant agar, because it has a very thick capsule. It has a very thick mucoid capsule. So first we'll see a short video about Klebsiella. Then we'll go to the PPTs, and then we'll move on to certain healthcare acquired or associated infections, hospital acquired or nosocomial infections caused by different organisms. So first let us see the video on Klebsiella. And by that time your friends will also arrive. If there is any issue with audibility or visibility, please let me know. So a lot of information was given in the video. If you want, we can share the link. It is available readily on YouTube. Okay. So as you understood, Klebsiella is a gram-negative rod. It has a polysaccharide capsule, and because of the capsule, it will appear as mucoid colonies. Because it is a lactose fermenter on McConkey, it will appear as pink mucoid colonies. And the other features, as you saw, it is urease positive, and then it can cause nosocomial infections. So a lot of diseases can be caused by Klebsiella pneumoniae, and because the name itself suggests that it can cause pneumonia, at the same time it is an important cause of urinary tract infections, bloodstream infections. It can also cause any other type of infections like meningitis, etc. So we'll discuss in short about Klebsiella. Already this has been taught in systematic bacteriology, but at the same time, this is a quick recap for you. And if you have any doubts, you can get back to us. At the same time, please go through the textbook, whether Bhavecha or Anantanarayan, whatever you are following. So it belongs to Enterobacteriaceae. It has been taught in the chapter of Enterobacteriaceae, where Escherichia coli, Proteus, and others are there. So there are different species like Klebsiella pneumoniae, Klebsiella ozeni, and Rhinoscleromatis, which causes rhinoscleroma. So it is basically a gram-negative non-motile organism, and because it is lactose fermenting, because McConkey medium, you might remember it was a pink colored medium, if you could just remember what you had already seen in your classes, practical classes. It was a pink colored medium, and the components were 5: plant, P for peptone, lactose, agar, neutral red, and taurocolate. And taurocolate is a bile salt, and because these are all Enterobacteriaceae, so that is why they can grow on McConkey agar. And because it has neutral red, whichever organisms will ferment lactose, because lactose is already a component, so whichever will ferment lactose, they will appear as pink colonies. Whichever will not ferment, like Salmonella or Shigella, those are non-lactose fermenters, those will appear as non-lactose fermenting pink, pale or light yellowish colonies. So pink colonies will be Escherichia coli and Klebsiella, whereas pale yellow or very colorless colonies will be Shigella or Salmonella. So these are lactose fermenting, and these are oxidase negative. Please remember, all the Enterobacteriaceae are oxidase negative. You might remember oxidase test was a test which we showed you. It was like on a filter paper which we used to put oxidase reagent on, and then once we smear the colonies, whichever is oxidase positive, like Pseudomonas, it will turn into a purple or a deep blue color. So this is oxidase negative. So these are all Enterobacteriaceae which are oxidase negative. These are all bacilli, so these are rod-shaped organisms. These are facultative anaerobes, which means that these organisms are basically aerobes. At the same time, these are facultative anaerobes. That means it can grow in the absence of oxygen also, but less abundantly. It is surrounded by a thick polysaccharide capsule, and in the olden days, when they did not have anything else, by seeing the capsule itself, which does not stain under the gram stain, you could know that these are Klebsiella. These are opportunistic human pathogens, which indicates that once there is some sort of catheterization or any other procedure which is done, these can cause infections in the hospital environment. So these are about 1 to 2 micrometers in size, and they have a thick surrounding capsule. You can see that this capsule does not stain, and because of the capsule, they produce mucoid colonies. So basically, it can be found as a part of the normal flora in the GI tract. At the same time, it can be found in the soil and water. But once there is a weakened immune system due to various reasons, once you give a lot of antibiotics, as we saw in the video, lots of carbaphenemases, ESBL, so those beta-lactamases, these enzymes which can be produced by these organisms. So whether it is beta-lactamases or whether these are carbaphenemases, they will destroy the antibiotics, which is why they develop antibiotic resistance. So they will have antibiotic resistance once you give antibiotics for a very long time to any patient. Maybe he's admitted, maybe he's there in the hospital for a long time. Naturally, if they get infected by these organisms, it is very, very difficult to get rid of these infections unless you start tetracycline or colistin or any other higher antibiotic. And beyond that, we do not have at present any other antibiotic. So we have to be very, very careful while starting antibiotics. So there are different types of infections which can be caused by Klebsiella, like urinary tract infections, pneumonia, septicemia, nosocomial infections, soft tissue infections, surgical site infections. So any type of infections which can be in the hospital, all of them can be caused by Klebsiella, and Klebsiella is especially problematic because of its antibiotic resistance. So via respiratory tract, which causes pneumonia, it can be spread, it can be enter blood. Once they give a central venous catheter or any catheter in which it is there for a long time. So maybe it is there in the hospital. So maybe from an employee or any other patient. So these this disease by Klebsiella can be spread. So it is spread very easily and rapidly, not through air, but the healthcare settings are the most vulnerable to Klebsiella infections due to the nature of the procedures that allow easy access of the bacteria into the body. So patients who are on ventilators, actually we have done a study here in Ashram also on ventilators, and the commonest organism which we found was ventilator associated pneumonia was due to Klebsiella, and many of them were antibiotic resistant also. So that is why we have to be very, very careful. So ventilators, even though you try to sterilize them, it is very, very difficult to get rid of these organisms which are very, very resistant. So catheters or surgery wounds, so these are highly prone to catching this Klebsiella infection. So traditionally what we have seen is respiratory tract infections, bloodstream infections, UTIs. So all of them can be due to Klebsiella. At the same time, different emerging infections can also be due to Klebsiella, like Lemierre syndrome, pyrogenic liver abscesses, antibiotic associated hemorrhagic colitis. So all of them can be due to Klebsiella. So meningitis, epidural abscesses, endophthalmitis, pneumonia, bacteremia, liver abscesses, necrotizing skin or soft tissue bone infections, all of them can be due to Klebsiella. And once it is not cured by antibiotics, or if the strain that is there in the patient is antibiotic resistant, it is very, very, very difficult to get rid of this organism. So different symptoms can be there in different infections depending on where the infection is located. For instance, meningitis produces the hallmark symptoms of bacterial meningitis, including fever, confusion, neck stiffness, sensitivity to bright lights. All those things will be there. Suppose there is bloodstream infection like bacteremia or sepsis, there can be fever, chills, rash, lightheadedness, or altered mental states. And pneumonia can be there, like fevers and chills. What will happen is, basically why we are telling these applied classes is that you will actually have an applied exercise in your final practical examination. There you will be given a question that will be based on a patient. So such and such patient was admitted due to such and such thing, like for example pneumonia, and then you collect a sputum sample and then you bring it to the lab, and then from the sputum sample you have grown this mucoid organism. So you will be given the blood agar plate, you will be given the McConkey plate. So on the blood agar plate, on the McConkey plate, you will see the mucoid colonies. Then you will be given the slides, you will see the organism under the gram stain. There will be a gram negative organism which you will see under the gram stain. Then you will go to the biochemicals. In the biochemicals, you will see it is urease positive, it is oxidase negative. So all those things you will see, and then you will come to the conclusion that it is Klebsiella, and all those things you will draw, you will write down, and then you will come to the conclusion that this is Klebsiella. Even the antibiotic sensitivity plate, that is a Mueller-Hinton agar plate, will be given to you, on which antibiotic discs will be placed. Some antibiotics it will be sensitive to, some will be resistant to. So you'll give a final report. That is, this is the organism isolated from this particular sample was found to be Klebsiella pneumoniae, and it was sensitive to such and such antibiotics, whereas it was resistant to such and such antibiotics. So in applied part of your practical examination, basically you'll answer the questions like this. So you'll draw all the things in your practical notebook, that this urease, as you have seen in your practical classes, the Christensen's urease medium, in which as you saw in the video, it will turn from light yellow to pink color. So you'll know that it has split urea into this urease enzyme has split urea into carbon dioxide and ammonia, and because of ammonia this phenol red has changed into a pink color. Okay, so these are the things which basically you will do in a applied practical class, which is not possible due to these virtual classes, but still whatever is possible, like what types of diseases, what types of symptoms that you will see, and what type of plates you will see, so those we can show. So basically you will have like the patient will have fever and chills, flu-like symptoms, the cough, which may produce mucus that may be yellowish or greenish or bloody, and there will be breathing issues. And the basic symptoms of pneumonia will be like high fever and chills, there will be skin clamminess, and in the lungs there will be sputum production, there will be pleuritic chest pain, there will be hemoptysis, there will be shortness of breath, there will be muscular fatigue and aches, and there can be joint pains or nausea, vomiting, high heart rate and low blood pressure, at the same time headache, loss of appetite and mood swings. So all of these can be there due to Klebsiella pneumoniae. And as we saw in the video, there can be different virulence factors also. As you might remember, there were the pili, short hair-like structures which were there, which are responsible for adherence or adhesion to the surface. Then there is the polysaccharide capsule, and as you saw in the video, there can be siderophores that have iron capturing ability. So these all are responsible for the virulence of Klebsiella. So typically in community acquired pneumonia, there can be acute infection of the lung pneumonia, which is acquired outside of a hospital or less than 48 hours after hospital transmission. And there can be many causes of community acquired pneumonia, out of which Streptococcus pneumoniae or pneumococcus is the commonest one. And there can be pleuritic chest pain, fever, dyspnea, tachycardia. So bronchial breath sounds and crackles on auscultation, and these things are seen on community acquired pneumonia. But in hospital acquired pneumonia, it is acquired after transmission, transmission occurs in a hospital after the patient is admitted into the hospital. And because these organisms are highly resistant to antibiotics, because they can produce different enzymes like beta-lactamases and carbaphenemases, so that is why the class of antibiotics which are beta-lactams or carbapenems, they cannot kill the bacteria. So because these are also a part of Enterobacteriaceae, they can also transmit these genes which are responsible for antibiotic resistance to other organisms also, which is a cause of concern. So because it is a common inhabitant of the normal microflora, once the patient is there in the hospital for a very long time, they can also acquire antibiotic resistance, and outside of the human colon, serious infection can occur. So as you already know, they possess a polysaccharide capsule. These are lactose fermenting facultative anaerobes, and different specimens can be collected depending on the particular disease, like sputum or urine or pus or CSF or even blood. Suppose it is a bloodstream infection, you have to collect blood. So on gram stain, you'll see. So I hope you remember that you have actually seen these things in classes. You have done gram stain, you have done Zn stain. So all these things you have done. Please try to remember these things on and off. Please go through different videos which are available on YouTube, different images that are available on the internet. So these are gram negative rods, and these will be very small actually. These are sort of very small gram negative rods, maximum 1 to 2 micron in size. And you can see that the capsule does not stain under the gram stain. So you can see that an empty space is there under the gram stain. If you look very closely, you can see that this is the polysaccharide capsule. So this is the polysaccharide capsule. So it is not stained by the gram stain. So on culture media, as you might remember, these appear very mucoid, very mucoid. Once you see the plates, the blood agar plate will be given to you, the McConkey plates will be given to you, nutrient agar plates will be given to you. Even these might be given as spotters. So that is why these classes are very important. Please try to attend these classes. Go back to the textbook and see all these images. Go through the written text which is there, which is actually very short. It is not very long. You will take hardly 15 minutes to go through it, but after every class is over, make it a point to go through. Especially I'm requesting boys, because I know girls will definitely go through. So these are very much mucoid in appearance. That is because of the polysaccharide capsule. Please remember this. Please do not forget these are pink colored mucoid colonies which are there. These are because this is because of lactose fermentation, and you can remember this. Once the plates are given to you, only on spotters you can get McConkey plates on which Klebsiella has been streaked and Klebsiella has been grown. So you can see these mucoid colonies, and you can write down that these are lactose fermenting pink colored colonies which are found on McConkey medium. Please remember this. This is a light pink colored medium, as you can remember from your practical classes. Do not forget these things. So a metallic green sheen can be seen on Embryoguard. We do not give you Embryoguard. So this is just for your information. So on KLED medium, which is usually done for cysteine lactose electrolyte deficient medium, which is used for urinary tract infections. So urinary pathogens, usually there also you can see mucoid colonies. This medium is also not given to you in practicals. This is also for your information, I'm telling you. So you can see that because of the mucoid colonies, you can see that if you touch it with a loop, you can see the polysaccharide that much, so much amount of the polysaccharide mucoid colonies which are there. So if you do an India ink capsule stain, India ink is nothing but a dark color background will be there, and because these capsules do not take up the stain, you can see empty spaces where the capsule is there. So this is an India ink stain. So usually we do all this India ink staining usually for CSF. So you might remember Cryptococcus is also having a that is a yeast, a true yeast, which is having a polysaccharide capsule. So for that also we do an India ink stain. So there also you will see the yeast cells where a clear area where the polysaccharide capsule is present, that is known as the where you will see that has not been stained. So here also in Klebsiella, because it has a polysaccharide capsule, so that is also not stained. Okay. So these are different antibiotic tests which are done. So these are test panels which are there. These actually will not be given to you. You will be given the tubes. I hope that you remember how you have seen oxidase tests and indole test. Indole test in which in a tube where pink colored ring used to form, that was in Escherichia coli. So Escherichia coli is indole positive, whereas Klebsiella is indole negative. Both are in lactose fermenters, but the Escherichia coli will be indole positive, whereas Klebsiella will be indole negative. So this is citrate positive. You might remember citrate, in which citrate one blue colored tube used to be there. You might have seen it there because it is bromothiamol blue. So one citrate is positive. Citrate is used as the sole source of carbon by certain organisms like Klebsiella. So Klebsiella will turn the dull green colored tube into a bright blue colored tube. So that is citrate positive. So urease positive and citrate positive, indole negative. Please remember at least these three tests. Please remember, indole test is negative, citrate test is positive, urease test is positive. So these three tubes will be given to you in your applied classes. So whichever infections are not drug resistant can be treated by normal gram-negative antibiotics, but those which are KPC producing, that is Klebsiella pneumoniae carbapenemase producing organisms, so you have to give a laboratory test results. So antibiotic sensitivity plate will also be given to you. So in those you can see what antibiotics are sensitive and what are not. Okay. So thank you very much for your attention. So we'll see two short videos on hospital acquired infections, after which we can discuss. Please go through the textbook. Do not forget to go through the textbook. You'll see the pictures, you'll see the text, so that you will remember whatever you have been taught. Okay, all these things have been taught to you in introductory. This is just a quick recap so that you can remember. Thank you so much for joining us today. I hope that you and your families are all safe. Please take care. Go through the chapters. Whatever applied chapter is taught, those chapters have already been taught to you in the theory classes. So please go through the same chapters. Go through and revise, because the exams and whatever you have to write down. So make it is to draw the diagram, especially in parasitology, and try to remember which are lactose fermenting, which are not lactose fermenting, which are aerobes, which are anaerobes. So all those things, please try to remember. Systematic bacteriology can be very confusing. If you have any doubts, you can always ask. If you need any reading material or anything else, it will be always given to you. So stay safe, stay happy. Thank you.